Hello everyone. Today we are going to explore one of the most exciting and rapidly growing fields in modern biology, that is bioinformatics, and its application in genomics and proteomics. In the last few decades, advances in DNA sequencing and protein analysis technologies have generated enormous amounts of data. A single genome sequencing experiment can produce billions of DNA bases, while protein studies can identify thousands of protein in a single sample. Managing, analyzing, and interpreting such vast datasets would be nearly impossible without computational tools. The real power of bioinformatics lies in integrating information from both genes and proteins. By combining genomic and proteomic data, scientists can give a more complete understanding of biological system. This integrate approach supports precision medicine, biomarker discovery, drug development, systems biology, patient stratification, and personalized treatment strategies. So here we will examine each of these applications in detail and understand how bioinformatics is transforming the future of biological sciences. Let us begin with Genomics. Genomics is a study of the complete genome of an organism. The genome includes the entire set of DNA present inside a cell. This means genomics studies all genes together rather than focusing on a single gene. In genomics, scientists try to understand DNA sequencing, gene structure, gene function, genetic variations. DNA sequencing means determining the exact order of nucleotides in DNA. The nucleotides are adenine, thymine, guanine, and cytosine. Understanding gene structure helps scientists identify coding and non-coding regions in DNA. Gene function explains how genes control biological activities and cellular processes. Genetic variation is another major focus. Variation in genes can lead to differences among individuals, including disease susceptibility. Now the question arises, where does bioinformatics help in genomics? Modern genomics generates enormous amounts of biological data. It is impossible to analyze the data manually. Bioinformatics provides computational tools, databases, and algorithms that help scientists to manage and interpret genomic information. The major roles of bioinformatics in genomics include genome sequencing analysis, that is, bioinformatics tools to analyze large DNA sequence datasets quickly and accurately. Next is gene identification. It is a computational method which helps to identify genes within long DNA sequences. Next one is sequence alignment. Bioinformatics compares DNA sequences between organisms to identify similarities and differences. Next is comparative genomics. In this, scientists compare genome of different species to study evolution and conserve genes. Next is genome annotation. Genome annotation identifies the location and function of genes within a genome. One excellent example is the Human Genome Project. The Human Genome Project was an international scientific effort to map and sequence the entire human genome. Without bioinformatics, handling such a huge amount of genomic data would have been impossible. Today, genomics plays an essential role in medicine, agriculture, forensic science, and evolutionary biology. So in simple words, bioinformatics act as a bridge between biology and computer science to understand genomic information effectively. Now let us move to the application of bioinformatics in genomics. Bioinformatics has revolutionized genomic research and has provided several practical applications. The first major application is genome sequencing. Genome sequencing involves determining the complete DNA sequence of an organism. Bioinformatics tools help in organizing sequence data, removing errors, assembling short DNA fragments, and analyzing complete genomes. A good example is again the Human Genome Project. The project generated massive amounts of DNA sequence data which were analyzed using bioinformatics software. Today, next generation sequencing technologies can sequence genomes rapidly and at lower costs. Next is gene prediction. Gene prediction is the identification of coding regions within DNA. Not all DNA sequences codes for protein. Therefore, bioinformatics algorithms help to predict the exons, introns, promoter regions, and regulatory elements. Next application is comparative genomics. Comparative genomics compares genomes from different organisms. This helps researchers to understand evolutionary relationships, to identify conserved genes, and to study species diversity. For example, comparing the human genome with chimpanzee genomes helps scientists to understand the human evolution. Next application is mutation analysis. Mutation analysis identifies changes in DNA sequences. Some mutations can cause disease such as cancer, sickle cell anemia, and cystic fibrosis. Bioinformatics tools to detect these mutations quickly. This supports disease diagnosis and genetic counseling. Next application is personalized medicine. One of the most exciting application is personalized medicine. Different individuals respond differently to medicines because of genetic variation. Bioinformatics helps to analyze a patient's genome to design personalized treatments. For example, selecting the most effective drug which will reduce the side effects and predicting the disease risk. Personalized medicine is become a major future direction in healthcare. Therefore, bioinformatics and genomics not only supports research but also directly improves the human health. Databases used in genomics. A database is a large organized collection of biological information stored electronically. These databases stores DNA sequences, RNA sequences, protein sequences, and functional information. Let us discuss some important genomic databases. First one is GenBank. GenBank is one of the largest nucleotide sequence databases. It is maintained by the National Center for Biotechnology Information or NCBI. It stores genomic DNA sequences submitted by researchers worldwide. Scientists use GenBank to retrieve DNA sequences, compare genes, and to study mutations. Next is European Molecular Biology Laboratory Database. It is another comprehensive nucleotide sequence database containing DNA and RNA sequence information. Next, DDBJ, that is stand for DNA Data Bank of Japan. Together, GenBank, EMBL, and DDBJ exchange data regularly and maintain synchronized global nucleotide sequence databases. Now let us discuss tools of bioinformatics. The utilization of various databases requires the use of suitable search engines and analysis tools. These tools help to analyze biological data efficiently. First is BLAST. BLAST stands for Basic Local Alignment Search Tools. It is one of the most widely used bioinformatic tools. BLAST compares a query DNA or protein sequence against database sequences to identify similarities. Application of BLAST include identifying unknown gene, finding homologous sequences, and help in studying the evolution. Next is FASTA. FASTA stands for Fast All. FASTA is another sequence alignment tool. It performs rapid sequence comparison. Both BLAST and FASTA are extremely important in genomic analysis. These tools and databases form the foundation of modern bioinformatics research. Without databases, biological data could not be stored efficiently. Without tools, the data could not be analyzed properly. Thus, databases and computational tools are essential pillars of genomics. Now let us understand the role of bioinformatics in proteomics. Protein-related data is highly complex. Protein can fold into complicated three-dimensional structures. They can also undergo modification after synthesis. Bioinformatics helps to analyze this complexity. The major role of bioinformatics in proteomics includes protein sequence analysis. That is, bioinformatic tools compare protein sequences to identify similarities and evolutionary relationships. Next, protein structure prediction. That is, the structure of protein determines its function. Bioinformatics predicts protein structure using computational modeling. Next is functional annotation. Bioinformatics helps to assign functions of proteins based on sequence similarity and structural information. Next, protein interaction analysis. Proteins rarely act alone. They interact with other proteins inside the cells. Bioinformatics helps to study these interactions and cellular pathways. Proteomics is extremely important in disease research, drug development, biotechnology, and in personalized medicine. The major application of bioinformatics in proteomics are: first, protein structure prediction. It is predicting the three-dimensional protein structure by using 3D structure modeling. Bioinformatic methods can computationally model protein structure. By understanding the protein structure, scientists can predict the function of a protein, the study interaction, and can design the drugs. Next is protein identification. Protein identification is commonly performed using mass spectrometry. Mass spectrometry generates complex datasets. Bioinformatic tools analyze these datasets to identify proteins accurately. This helps in clinical diagnostics, disease research, and again in biotechnology. Next is protein-protein interaction analysis. Proteins interact with each other to perform cellular function. Bioinformatics helps to construct protein interaction networks. This helps scientists to understand the cellular pathways, signal transduction, and metabolic processes. Next is functional analysis. Functional analysis determines the role of proteins in biological systems. Bioinformatics predicts protein function using sequence similarity, structural motifs, and evolutionary conservation. Next is disease research. Proteomics plays a major role in identifying biomarkers. Biomarkers are biological molecules that indicate disease condition. For example, cancer biomarkers, diabetes biomarkers, cardiovascular disease biomarkers. Bioinformatics helps to identify these biomarkers rapidly. This supports in early diagnosis of a disease, monitoring of disease, and helps in therapeutic development. Now let us move towards some important databases and tools used in proteomics. As we already discussed, proteomics deals with the study of proteins, their structure, function, and interaction. Since proteins are extremely complex molecules, scientists require specialized databases and computational tools to analyze them efficiently. The important databases are first Swiss-Prot. Swiss-Prot is one of the most reliable and curated protein databases. The word curated means that the information stored in the database is manually reviewed by experts for accuracy and quality. Swiss-Prot provides protein sequences, functional information, structural details, and biological annotations. Researchers use Swiss-Prot because the data is highly accurate and well organized. For example, if a scientist discovers a new protein sequence, they can compare it with Swiss-Prot entries to predict its function. Next is Protein Data Bank. Protein Data Bank stores three-dimensional structures of protein and other biological molecules. As we know, protein structure is closely related to protein function. Scientists use techniques such as X-ray crystallography, NMR spectroscopy, cryo-electron microscopy to determine the protein structure, and these structures are then deposited into the PDP database. Using PDP, researchers can visualize proteins, study active sites, understand the molecular interaction, and can design the drugs. For example, during drug discovery, scientists study the 3D structure of disease-causing proteins from PDP to identify the possible drug binding sites. Next database is UniProt, another very important database. UniProt is a comprehensive protein resource that combines protein sequences, functional annotations, structural information, and evolutionary data. It is widely used in both academic and pharmaceutical research. Now let us discuss important proteomics tools. PyMOL. PyMOL is a molecular visualization software. It allows scientists to create detailed 3D images of proteins and biomolecules. Using PyMOL, researchers can rotate protein structures, zoom into active sites, can analyze molecular interaction, and can create publication quality images. It is especially useful in structural biology and drug designing. Next tool is BLASTP. BLASTP is a protein sequence comparison tool. It compares a query protein sequence against protein databases to identify similar proteins. Its applications include predicting protein function, studying evolutionary relationships, and identifying the homologous proteins. If two proteins are highly similar in sequence, they often perform similar function. Next tool is ExPASy. It is an online proteomics resource portal. It provides multiple tools for protein sequence analysis, structural prediction, for molecular weight calculation, and for protein characterization. Scientists frequently use ExPASy in routine protein analysis. Overall, these databases and tools are essential in modern proteomics research. They help scientists to store, visualize, compare, and analyze protein-related information efficiently. Without these bioinformatic resources, studying protein at such a large scale would be extremely difficult. Now, one of the most important applications of bioinformatics is drug discovery. Drug discovery is the process of identifying and developing new medicines to treat diseases. Traditionally, drug discovery was a very long and expensive process. which involved years of laboratory experiments, testing thousands of compounds, and high chances of failures. In many cases, developing a single successful drug could take more than 10 years and cost billions of dollars. This is where bioinformatics has completely transformed the pharmaceutical industry. Let us understand the role of bioinformatics in drug discovery. The first step in drug discovery is identifying a suitable drug target. The drug targets are usually proteins, enzymes, receptors, or disease-related genes. Bioinformatics helps scientists to analyze genomic and proteomic data to identify targets associated with disease. For example, if a specific protein is overexpressed in cancer cells, it may become a potential drug target. Then, virtual screening. Traditionally, scientists had to test compounds physically in laboratories, but now bioinformatics allows virtual screening. In virtual screening, computers analyze thousands or even millions of compounds digitally to identify promising drug candidates. This saves enormous amounts of time, cost, and laboratory effort. Next is drug design. Bioinformatics also support rational drug design. Scientists can design molecules specifically to interact with disease-causing proteins. This improves drug specificity, drug efficiency, and treatment effectiveness. Then molecular modeling. Molecular modeling predicts how biological molecules behave and interact. It helps researchers visualize the protein structure, drug-target interaction, and the binding sites. This improves our understanding of how medicines work at the molecular level. Next, predicting the drug interaction. Bioinformatics can also predict possible drug interaction, the side effects, and the toxic effects. It improves the patient safety and reduces failure during clinical trials. Today, bioinformatics is widely used in pharmaceutical industries worldwide. It has accelerated the development of drugs for cancer, infectious disease, genetic disorders, or neurological diseases. Therefore, bioinformatics has become an indispensable tool in modern drug discovery. Now discuss the major steps involved in bioinformatics-based drug discovery. This process is commonly called the drug discovery pipeline. Each step uses computational tools to improve speed and efficiency. First one is target identification. In this step, we will identify the biological target linked to a disease. Target may include protein, genes, enzyme, and any cellular receptors. Bioinformatics analyze biological datasets to identify these targets. For example, if a protein is essential for the survival of cancer cells, scientists may target that protein with drugs. Next step is target validation. After identifying a target, scientists must confirm that it truly plays a role in the disease. This process is called target validation. Validation ensures that targeting the molecule will produce therapeutic benefits. Without proper validation, drug development may fail later. Next step is lead compound identification. The next step is identifying the lead compound in the disease. Lead compounds are molecules capable of interacting with the target effectively. Using virtual screening and computational analysis, scientists can rapidly identify promising compounds from large chemical libraries. Next step is molecular docking. Molecular docking is one of the most important computational techniques in drug discovery. It predicts how a drug molecule binds to a target protein. Docking studies help determine the binding affinity, binding orientation, and the stability of interaction. A strong and stable interaction usually indicates a better drug candidate. Docking significantly reduces the need for costly laboratory screening. Next is drug optimization. After identifying the lead compounds, scientists modify them chemically to improve their properties. Drug optimization focuses on improving the effectiveness, stability, solubility, bioavailability, and safety. The goal is to develop a compound suitable for clinical use. Next is clinical trial support. Bioinformatics also helps during clinical trials. It assists in managing patient data, statistical analysis, biomarker analysis, monitoring treatment response. This improves the accuracy and efficiency of clinical studies. The benefits of faster drug discovery. The benefits of bioinformatics-based drug discovery include reduced development cost, improve the efficiency of the drug, better prediction accuracy, and high success rate. Now discuss some important computational techniques used in modern drug discovery. First one is molecular docking. Molecular docking predicts the interaction between a drug molecule and its target protein. This technique helps to determine how the drug fits into the target, the strength of binding, and the possible interaction sites. It is similar to fitting a key into a lock. A better fit usually indicates a better drug candidate. Docking studies are widely used before laboratory experiments because they save time and cost. Next is virtual screening. Virtual screening is the computational screening of large chemical libraries. Scientists use software to evaluate thousands or millions of compounds rapidly. The goal is to identify compounds most likely to interact with the target protein. Compared to laboratory screening, virtual screening is faster, cheaper, and more efficient. Next is QSAR analysis. QSAR stands for Quantitative Structure Activity Relationship. QSAR studies the relationship between the chemical structure and biological activity. Using mathematical models, scientists predict whether a compound may become an effective drug. If a compounds with similar structure show similar biological activity, researchers can design better drug molecules. QSAR reduces unnecessary experimental work. Next is artificial intelligence and machine learning. Artificial intelligence and machine learning are rapidly transforming drug discovery. Artificial intelligence systems can analyze enormous biological data sets much faster than humans. It includes predicting drug activity, identifying drug targets, designing new compounds, predicting toxicity. Machine learning models can improve continuously as more data becomes available. This is one of the fastest growing areas in pharmaceutical research. Next is molecular dynamics simulation. Molecular dynamics simulation studies molecular movement over time. Unlike docking, which gives a static interaction, molecular dynamics shows how molecules behave dynamically. It helps scientists to understand protein flexibility, stability of drug binding, molecular motion, and interaction changes over time. This provides deeper insight into molecular behavior. Overall, these computational techniques have revolutionized modern drug discovery. They reduce experimental workload and improve the efficiency of pharmaceutical research. Although bioinformatics has transformed biological research, several difficulties still exist. First one is massive biological datasets. Modern sequencing and proteomics technologies generate huge amounts of biological data every day. Managing this data is a major challenge. Researchers require powerful computers, high-performance servers, and advanced storage systems. Handling big biological data efficiently remains difficult.